Good morning, everyone. For this part of the workshop, we're going to um, summarize the group activity that we did with our in-person attendees yesterday. So first of all, I want to first thank and acknowledge the people from FDA who contributed to preparing these exercises and also leading the group activities. Dr. Ying, Jiang, Sam Raney, Priyanka Ghosh, Yang, Mengmeng, Megan Kelchen, Hiren Patel, and Ahmed Zidan. So thank you very much. Go to the next. Okay. We'll wait till, okay. So first, I'm going to give an overview about what the exercises were, and then we're gonna hear from a volunteer from each group, where they would summarize the discussions and the lessons learned, and so that we can give a summary for our online audience who did not get a chance to participate in these exercises. So we first introduced a hypothetical drug product. It's a topical cream indicated for plaque psoriasis. We provided some preliminary information about physical chemical properties of this drug, solubility, chromatographic method, and also provided a drying rate data to show that it undergoes metamorphosis and dries. Then we ask our participants to come up with the IVPT method for this drug product based on the information provided about this drug product and also based on the resources they have access to in their companies. We gave them some time to think about different method parameters or tests that would be applicable during method development. And then we discussed what they were proposing and what would be appropriate considerations during method development. For the next activity, we provided two donor IVPT profiles. And in one scenario, we gave these two profiles and where we didn't see or didn't get quantifiable levels in half of the replicates, and especially in the first 24 hours. And we asked them if this is the results you get from your first IVPT study during method development, what would you do? Do you continue with this method? Is it good enough, or you need to do some optimization? And if yes, what aspects or of method parameters you would modify? And for the second scenario, which was independent of the first scenario, these are the two profiles generated from the first IVPT study using the proposed method. And again, if this, based on these results, if the IVPT method is adequate to move forward, or if it needs further optimization, and if yes, what considerations we need to have for optimizing the method. Again, we provide them this method parameters list and ask them which one they would tweak and modify. Eventually, we provided some examples of optimized IVPT profile from a method development, and we want to ideally see, we want to see it at least in 3 donors, you have established an adequate profile, and making sure that your method is adequately developed and it's ready to move to this next stage of method validation. And we would discuss that, all these considerations and step-by-step documentation of these changes that you would apply to the method and the thinking process behind why you modify certain aspects to get to your optimized method. Then for the third activity, we discuss sensitivity study. If they have the adequate developed method and they want to do a sensitivity study, what would be their strategy? if they're modulating the dose amount, what would they select as a higher dose amount for the sensitivity study. We presented some hypothetical profiles from a sensitivity study between 3 different dose amounts and asked the group if they consider the method to be sensitive based on these results. Next, we discuss selectivity study and what would be the strategy for creating an altered formulation that can provide a distinct bioavailability compared to the RS. And again, an example of a selectivity results and whether based on these flux profiles and these cumulative amount profiles, they consider the method to be adequate, selective or not. And at last, there were some examples of an adequately selective method or acceptable selectivity profile given. And the fourth activity, I'm not sure if all the groups got to the fourth activity or not, was just an example of, okay, imagine you have developed and validated a method for a cream product. Now, next you want to develop, next you want to have the method for an ointment product for the same API. What would you do as part of method development strategy? So with that summary, I'll invite.
So for the yellow group, we have Katerina and Vishal doing the debrief for us. So we'll start with Katerina.
Hello. Okay, I will start with the importance of the choice of analytical method. So, and the sensitivity of the analytical method. So the properly selected and developed analytical method will show you information whether you can achieve the sensitivity of your tested product. And for that, so the draft guidance, it recommends using either the mass spec method or the HPLC UV method. But like in most cases, when you have the low strength product, the MS is more sensitive. But yes. But regarding that, we also discussed that here could be several challenges. For example, you need to properly clean your sample to get rid of any surfactant if it was used in the sample in the receptor solution. Yes. So I don't know, I can go further with, for example, second method development parameter, for example, the receptor solution composition. So here the solubility of the active ingredient should be considered. For example, we had the API that wasn't soluble in water and was soluble in DMSO and ethanol. So it left impression that the organics could be also included into the receptor solution. However, it's better not to use it. And if it's still if the organics or like DMSO was selected in the receptor solution, it should be justified, and it should be shown also that the skin barrier integrity is not compromised. And still, the FDA guidance recommends not to use any organics, and it's also makes sense because there is no organic solvents in the physiological solutions. So, based on the solubility testing of the API, it could be also concluded that what receptor solution could be used, whether it be just pure the PBS solution or any other surfactant could be added to increase the solubility of the API. But also not more than 6%, because it can also impact the sample itself, and it could be very difficult to clean it and to inject into the MOSPEC.
Hi. To add some of the points what we discuss. So first, initially we got this information. It has the log P information, pK and solubility data and the drying rate data, uh, plus molecular weight. So based on that, we discuss about the property of the molecule, where being log P 4.5, it is lipophilic molecule and water insolubility. So the first challenge would be to decide the receptor media. So we discuss about the we should start with PBS plus some amount of surfactant, and we discussed that only 20 is the preferred one. One of the reason for selecting the only 20 is previously that data has been published that up to 6% of the only 20, it is not impacting the skin integrity. So we thought of starting with those receptor media, and for the first study we decided we should go with 96 hours to have the full profile and to understand whether we are getting the Tmax and the decline phase and the increment phase or not, and based on that subsequently we can further optimize the method. And yes, so for the first experiment we discussed to go with the sample weight of highest, which is 15 mg per centimeter square. If we go to the next slide, where there are two data. So we discussed that there are 2 points. One is the variability in the two donors, and the other is very low permeability observed in the remaining four replicates. So we discussed that how to overcome this challenge. So for that, we discuss multiple things. One of the thing is to alter the receptor media. Maybe if the saturated solubility supports, then we can reduce the receptor volume in order to get the sensitivity. Plus, we discuss about improvement the analytical technology also, so that can provide us the more reliable data. And we discussed that we should control the dosing technique in order to avoid the outlier kind of outlier what we have observed here from two of the cells. So the another thing what we discuss is for the selection of surfactant if we required. So we discussed that yes, FDA is okay to have the different surfactant than only 20, considering that we prove that the skin integrity is not impacting. So we discussed there were some suggestions to use the DMSO and propylene glycol. So again, if we prove that it is not impacting the skin integrity, then and if we justify it properly, then it might be still okay, but strong justification is required for that. And how to go to next? Do you know how to go to next slide?
Yeah, I think we can.
Yeah.
Have a couple of comments.
Yeah.
Mm-hmm.
So quickly I'll go through. So based on this slide, we discussed that there is no decline phase. So the options came is to increase the IVPT duration, which is right now in this one it's 48 hours. We can go up to 96 hours, and if we see whether we are getting the decline phase or not. Another discussion point came is the wipe off. So that it has pros and cons. So if we have a very good skill is required if we want to adopt the wipe up technique. So that was what we discussed. And quickly for the sensitivity, we discuss about major two techniques. One is if we have, let's say, if we go with the 96 hours and if we are getting the complete profile, then the one of the option to go the sensitivity is to have the three different weights. So initially we thought of going with 15 mg per centimeter square. So we discussed that we can go 5 and 45 to start, so 3 times low and 3 times higher, and we'll see whether we are achieving the sensitivity or not. And for the wipe up technique, we discussed that let's say if we are in our method we have finalized to wipe off at 24 hours, then maybe we can do wipe off at three different time points and we see if we are getting the desired profile or not. So you can add more.
Yeah, I can add here that for the sensitivity study, if we select the modulation of the dose amount, we need to also, so one option is to also see the drain data. So if our drain data shows that, so the drain data, if we use different amounts of the cream of the product, should have the same profile. And if the profile is different, we cannot use the higher amount of the product. So, and also we discussed that it is a very easy study that could be conducted prior of our initial IVPT experiments when we select the initial dose amount. So we just evaluate the how our product is drying, just using the balances.
So thank you so much to our entire group and specifically to Vishal and Katerina for offering to speak. I just wanted to say that the most interesting part of the discussions for us, I think definitely DMSO was placed as a trigger on the first page of this document. And we had some interesting conversations for using DMSO in the receptor solution, and mostly related to how to qualify a solubility modifier. But I do want to clarify that I do not recommend using something like DMSO or ethanol in the receptor solution, because that is just creating unnecessary challenges for us. And then the other aspect that I really found interesting during the discussions is how a quick and easy drying rate study can really help us to select doses for sensitivity, or how an understanding of the microstructure of the product and drying rate can help us to select products for the selectivity study. So I'd like to thank Mengmeng for being an excellent co-activity lead for this, and turn it over to the next group, which is, I think, Tannaz's group. Tannaz, you would like to go next?
Thank you. I invite Aditya to come and summarize our group discussion.
Thank you, Tannaz. So yeah, I think group number 2.
Advance the slide. You need to. Yes.
So the previous group has extensively covered the initial points, but I'll just quickly go over for the first method. I mean, the first data set which was presented. I think the analytical method, the mass spectrometry and receptor solution, as they mentioned, will require the solubility, I mean, the permeation enhancers like oleate. So anything different from oleate per guidance needs to be justified. Equipment, choice of equipment, we propose the low-gun diffusion apparatus, which involves complete media replacement. And since it's a very low permeating drug, source of the skin, any dermatome skin should be evaluated from any anatomical region for screening. Barrier integrity assessment, 12 value, and dose amount selection anywhere between 10 to 13 milligram per centimeter square. And study duration, we propose 96 hours. Initial assessment can be performed, and based on the data available we generate and we can always change the dose duration, I mean the study duration. And accordingly, the sampling frequency can be adjusted. For the second set, since we observed very low permeation, we thought of checking with the alternate permeation enhancer, which apart from oleate like bridge 20 or something, or if anything apart from the guidance recommendation as stated before, we need to adequately justify. It doesn't impact the barrier integrity. And accordingly, anything we change in the receptor media, we need to evaluate the solubility and receptor solution stability to ensure that the analyte is stable and data is the actual representation of the permeation. And we can check the dose IVPT study duration for like 96 hours as to see the initial flux profile, and accordingly we can adjust. The third data set which we observed was, sorry. Yeah, so for this particular data set, how to improvise the flux profile, we saw, like as they stated before, we can change the study duration from like 96 hours to see if we see a rise and subsequent decline to check. And once we get that, we can accordingly improvise the flux profile. Alternate option, as stated before, is the wipe off technique. But anything we do wipe off, as we discussed before, needs to be adequately justified and it doesn't impact the study outcome. So anything we can do the wipe off, but it needs to be properly scientifically and data driven justification should be provided. So accordingly, we can improvise the methodology. For the subsequent data set, also is for the sensitivity study. So what we observed is that the method which the data set which is presented, few of the donors, like 2 donors, demonstrate some difference in the flux profile for the different dose amount, but like 3 other donors are not qualitatively discriminating. So we cannot conclude the method is sensitive at this point. So in order to obtain the discrimination, we need to have the proper modulation of the dose amount. So anything like 3 times the dose from the target dose should be selected and evaluated. If that demonstrates, or we can obtain an adequate discrimination qualitatively based on the selection of the dose amount. So this should be evaluated as a starting point to see if we can obtain a discrimination, sufficiently discriminative flux profile across the dose amount duration. So for the next data set was the selectivity. So if you can see the data here, we are not observing the sufficient selectivity or the discriminatory flux profile from the altered dose, from the altered dose from the control dose. So in order to obtain an acceptable selectivity flux profile, what we can do is we can change the product matrix. I mean, anything, if we are evaluating a cream, we can check if we can select a sufficiently acceptable altered dose like the ointment, which has different product matrix having different excipients that alter the permeation of the analyte across the skin. So accordingly, we can select and see if that demonstrate a sufficiently acceptable discriminatory flux profile. So the next data set which we see here is regarding the selectivity, if this data set represents the sufficiently selective drug flux profile. So if you can see over the qualitatively, if we can see the altered dose, which is represented by green color, when we are plotting against the standard error bars, we are seeing sufficiently discrimination. So individual replicates, which represents that the individual replicates are sufficiently discriminating from the control, that is the test and reference dose, which proves that the obtained IVPT profile demonstrate the discrimination against the altered formulation. So we deduce that the obtained, the presented flux profile is discriminative and selective. And finally, the next question, the last question was that if we are presented with all different formulation having the same molecule but a different product, I guess. I think that's what the question is. So how would we proceed with the IVPT method development? So since we have already have a baseline method for, let's say, if we have a cream method for a particular molecule, and if we are targeting to devise a method for an ointment, so how would we proceed? So we'll take the baseline method for the cream, and we can run the IVPT profile as the starting point. What improvisations can be made is in terms of the dose application and the dosing duration for designing the IVPT study. And also the sampling frequency can be altered and the IVPT study duration, because let's say if we have an ointment, it may have a different drying, I mean, the rate of drying would be different as compared to the cream. So metamorphosis would be different. So accordingly, we have to improvise in terms of the dose application and the dosing frequency and the sampling frequency, as well as the IVPT study durations. So alternatively, the receptor media and everything, we don't need to significantly alter considering the baseline method on hand, and accordingly we can proceed for the next IVPT development stages.
Thank you very much for the nice summary. A few points that I want to add is I think we discussed that any of the equipment, IVPT equipment that has been introduced in this workshop would be acceptable. We don't have a particular preference. It seemed like within our group, most people had access to Logan more than the other equipment at their side. However, it also depends on because you want to also modulate, if possible, volume, the active surface area to improve the profile if you have a low permeating drug. You may not be able to do that with every equipment. Also, we discussed about barrier integrity assessment test, and some folks shared their experience that, for example, with TIR, it's more difficult to optimize it, but it can maybe more sensitive than TWL. However, with all the equipment, you cannot conduct TIR with all the equipment. For example, if you have an automated inline Logan cell, you cannot use TIR device with that. So it depends what you have access, and then which parameters you can modify and optimize. You see. Also, another consideration is while you can, for example, for some of this equipment and the cells, you can use bigger magnets to adjust the volume. But if you have surfactant, you need to consider that using bigger magnets may cause creation of more bubbles or micro bubbles that can impact your data. And for the I guess another thing I want to note, and then I'll give the time to the other group, is for this selectivity profile example, is, as you see, we have a lot of variability in the data. The standard deviations are overlapping. And while here we are showing the mean profile and the mean donor profiles, not the individual replicates, but we see at some time points, especially earlier time points in donor 1 and 3, we may have aberrant data that contribute to the shape of the profile. So like if you have results such as this, the issue may not only be the altered formulation. The issue in addition to that, maybe you have variable data, so you need to make sure you have adequate control in place and further optimize your method to reduce the variability in order to establish good discrimination. Thank you. So I'll invite group red next. Thank you.
Hello. So we were the group red, and Lakshmi and Ross are going to summarize our discussions.
Yeah, I'm going to start off and then let Ross take over. Primarily, what we discussed was first page. You look at the drug substance and drug product characteristics. What's the molecular weight? Log P is 4.5. That is highly lipophilic molecule. The pKa is 9, so it's a basic compound. And we looked at the drying rate and the other excipients that are used, like the solvents that are used. So one thing we realized during the discussion early on is because of the presence of ethanol, you may have a kind of a faster profile, which you can expect at the beginning. So that was the one. So I just want to go over the initial method, and then I will hand it over to Ross. So just like the other group members discussed, we look at the sensitivity of the method, whether to go for LCUV or a mass spec, depending upon the sensitivity that you need. The receptor solution composition, again, we talked about the surfactants, use set at 20 or BSA, and also using gentamicin sulfate or the as antimicrobial agent in the receptor solution. Initially, the tests you would conduct for the receptor solution or solubility, of course. We also talked about the stability of the receptor phase itself, and then also to see if any of the surfactants you use are compatible with the receiver phase that you want to use. Source of skin, we again, like anyone else, we talked about the differences between cadaver skin and surgical procedure skin. So obviously, cadaver skin is easily accessible. The barrier integrity assessment, we looked at the TEWL as well as TER, discussed at a high level, didn't go in detail. The dose amount for the first IVPT study would be, as per the guidance, 13 to 15 milligram per square centimeter. The application technique we discussed in a bit of detail, a dose application technique, either glass rod or a HPLC vial or a capillary, and discussed the various options. The study duration, we looked at suggestions between doing a shorter duration and a longer duration. And then we discussed that about 96 hours to start off with probably a fewer sampling frequency to just gauge what the first IVPT experiment will look like. And then the dose duration, we decided to keep it as the same as the study duration for the first experiment. And the sampling volume, again, we discussed it, and whole receptor phase volume replacement is what we discussed. Here to Ross.
Thank you, Lakshmi. The only thing I would add there is that for several of these method parameters, a lot of our group discussion focused on trying to find whatever the best option is that's repeatable for your lab and that is well established already as a method in your lab. So whether it's transepidermal water loss or TER, doesn't really matter, or whether it's spreading with a glass rod or pipetting or using the HPLC top in order to spread the application. Whatever you can do that is going to be the most dependable, repeatable, that's really what you should reach for. So on the next activity, the second activity, optimizing the method results from the first study, we saw these flux profiles that were not increasing early enough in the study. So we were getting basically zero flux for the first 24 hours of these studies. And most of our discussion here focused around trying different methods to increase the concentration gradient, just to increase that diffusion drive for the API in order to get to a J-max that's going to be apparent within the first 24, 30 something hours of the study, and then allow us to actually see that declining flux that we need to see. So a couple of ways that you could go about doing that. You could, of course, increase the dose. You could decrease the volume of the receptor. You could try to increase the length of your study from about 50 hours to 96 hours, just in case you're missing that tail. And then for the next activity, we largely agreed with everything that the other two of our groups have already suggested. This was the study that we were trying to look at the scenario 2 and discriminate between these different donor A and donor B flux profiles. So here we suggested potentially decreasing the dose as the first step. And again, consider extending the study, because we're not seeing, for example, in that first donor A profile, we're not seeing that decline in flux that we want to see toward the end of the study. Consider extending the study, or consider doing the wipe off. And then for a sensitivity study, as the other groups have already noted, the main issue here is just that high and medium dose, or the target and the high dose. You've got these error bars that are essentially crossing each other, so we're not really significantly discriminating between the high and the medium dose. So you can try to increase the dose of the high product. So from 20 to 30, that was pretty much our group consensus that would be by far the easiest first step. And then for the selectivity, we largely agreed completely with everything that's already been suggested. Try different, any different vehicle based formulation. So cream to ointment, easy enough approach to solve this issue. And then we didn't get to the last activity because we had a robust discussion about the others.
Thank you, Lakshmi and Ross, for summarizing our discussion. A few additional points that I wanted to make. When we discussed the receptor solution, we had an interesting discussion about the pKa of the drug product and whether we could modify the pH of the receptor solution, or if that we can do that in combination with adding solubility enhancers. Some things that we discussed when we consider that is compatibility with the skin and compatibility with the API. And we didn't really come to a consensus necessarily on that, but that's one thing that we discussed that I encouraged everyone to come in as a pre-ANDA meeting if they wanted to discuss that more. Another thing that we discussed is when we looked at scenario 1, where we had limited permeation at the beginning of the profile, we talked about how one thing you could look at is making sure that your dose or your skin mounting procedure and your dose application procedure are optimized and are consistent and reproducible, and it's not adding unnecessary variability. And then the final point I'll mention is when we talked about scenario 2, we talked about the potential, or scenario 2, where we did not see the decline in permeation in the elimination phase. One thing we talked about was the use of a wipe-off, and if you wanted to use a wipe-off, you really need a consistent and optimized method. But if you extend that study duration, you may not need to consider that wipe-off, and we heard the panel discussion this morning about that aspect. And the other thing I'll mention is that, well, Ross mentioned that we don't, that we may want to consider decreasing the dose. That's one thing that you may want to look at the study duration first, and you want to make sure that if you would decrease the dose, that you're not going below the recommended dose of 10 milligrams per centimeter squared. So thank you everyone in group 4 for the great discussion, and thanks again for summarizing.
We have some good news to report. We found Hader. And so I'll let him jump into it.
I tried to run away, it didn't work. Hi, everyone. We had an amazing discussion yesterday about this. So the discussion started just like, what will you do if you get this molecule, this challenge with cream? You know the molecular weight. So our first impression was like, okay, molecular weight, that means 250, it's within our limit, so it should go through the skin. What having high log P would limit the permeation into the skin. And then we looked more into the solubility. So what kind of receptor solution can we pick for such a molecule? And there was a lot of chat about whether we can add ethanol to the receptor solution. And the simple answer is no. The guidance, there is a potential impact, not potential, there will be an impact on the skin integrity if you add ethanol to the receptor solution, which could impact your results. So the only solution for us moving forward was to use the OLITH 20, and we accepted that we're going to use the range of 0.1 to 0.2. The pH of the receptor solution came back again, just in case that if we lower the pH, and what will be the range acceptable. We agreed 7.4, like the normal pH is fine, as mentioned earlier. If we want lower, we need to go to the FDA to ask them. But if it goes down to 7, may be useful, but might be acceptable, but that's something needs to be confirmed with the regulatory agency. Testing solubility, that's the first thing. To qualify your receptor solution, you need to look into the solubility, saturated solubility, especially at the temperature that you run your study at. So 34 degrees, 35 degrees, even better. And then that will reflect your actual data, also the stability at that temperature too. Equipment, as mentioned, start with what you have and then move forward. Type of skin source, that was also a big discussion because we agree that dermatome skin is the golden standard, but you need also to understand where it's coming from. Is it being harvested properly? Is the skin managed properly? Do you know anything about, again, if it was treated with glycerine, if there are any damage to the skin during the harvesting process? Methodology with the barrier integrity, again, that's been talked a lot in these discussions. So tool is the golden standard. There are a lot of, actually there's we didn't test it, but there's a publication with Howard Myback where they compared the 3 instruments. That's something that could be interesting. And the dose amount, we said because of the low permeation, we're going to start with something high, something between 10 and 15, and we're going to start with a longer study duration as much as possible. And in terms of the dose, we start with a longer dose duration too, to maximize the amount of API going into the receptor solution. Receptor sampling volume, and that will be also the smallest possible, just to get the highest amount of API. Okay, can we use this? Okay, that's fine. No worries. I was trying to get to maybe, no. This one? Okay. So then we get to this challenge. So where do we start when we have a problem like this? So the simplest way, if you don't want to touch the IVPD, start with an start with the analytical method, improve the sensitivity. Let's see into the picogram level and see what kind of result you will get. Because the main interest is the donor on the left, donor A, you still see flex max appearing for the remaining cells. So that could be an improvement point for you. Another point that was mentioned, use a larger cell, larger surface area. Hopefully that will help into improving the amount and the receptor solution. And as we move forward, we looked at this profile. So the main point is you still see flux max in one of the donors, the other one you don't. So you can either go for a longer duration, which is that's simpler. You're not going to change your method. And you can, as other people mentioned, maybe wipe off, but I would say start with a simpler one. As we moving forward, I will go to this one. There was a big discussion about this because to find the sensitivity properties, again, the focus was use threefold dose, whether higher or lower. Start with something, a lower dose is about 2 milligrams and a higher dose in the range of 25 to maximize your sensitivity, because the expectations is to get a full separation between them. And I think that also mentioned in the previous discussions, because you see in a couple of donors here that the separation was not fully established between the other donors. There was another point that was mentioned, like, okay, but it's, going back. Although that you see some separation at the end, that doesn't solve the problem. I think the expectation is to have a full separation during the whole the process. And I think next one is this. So the control here is the alternative formulation is higher. So it's not as a negative control. And as mentioned, that the test could be an ointment or any other product that available within the same parameters that you're looking for. So this was not enough to show the selectivity. And as we move, I think we stopped here at some point. I think we ran out of time. The discussions were extreme. And at the end, I think that's, as mentioned earlier, is an acceptable profile. And I leave the floor to the boss.
Not the boss. I think Priyanka is the boss. Thank you, Hiren. That was an excellent synopsis of a really good conversation we had in the room. I did want to comment, maybe clarify that as relates to this this part of the exercise. The conversation was an observation that here, when we're looking for sensitivity, it's ideal if you can show lower and higher sensitivity. But the lower part, really, we know the method can measure lower permeation because it's measuring lower permeation all the way up to there. The question is, are we maxed out where a product that is different on the higher end would not be able to be discriminated? And so it's really being able to show that higher part that's important. Now, if the time points all overlapped in the very first few, separated nicely, nice separation across a few points of Jmax, and then came back toward the end and then overlapped again, I would say that's a very good demonstration of the sensitivity of the method. The point being that you do not have to have separation between the profiles at every point along the profile. And so then that being the principle, well, is this acceptable? In this case, there's not separation in the early part, but in the later part of the profile, beyond the error bars, there is separation. And I think that one has to be open to saying, if you've made reasonable, and this is why the method development experiment in the report is so valuable, to be able to demonstrate that you have a good understanding of the behavior of the method, it's well characterized during method development, and reasonable efforts have been made to optimize the method. And the behavior of the product may be such, and we've seen this even, for example, with metronidazole gel and cream, two very different products with very different bioavailability, and yet in the early time points right up to Jmax, they essentially overlap, and then the cream keeps going, and the gel comes down. Now, I think that's a very reasonable demonstration of the ability of the method to, kind of be sensitive to those differences in bioavailability between the gel and the cream there. And so the recommendation here was, well, not sure that this is really a fully optimized method. It's probably worth a little bit more effort to try to optimize this, and perhaps to try even, a bigger spread in the doses, because you could, I would try personally to try get data a little bit better than this. But I think in principle, we need to kind of be open to understanding that you do not need to show separation at every point along the profile in order to be considering it, a well-discriminated profile. The other point that also came up is, what if the nature of your formulation, let's say the reference formulation, is such that a lot of development work at that time went into maximizing what can be achieved in terms of permeation through human skin. And you really can't get any higher permeation than that with some alteration to the formulation, because someone who is really good at formulating the reference product has already kind of maxed out what's achievable. Adding more concentration of drug or no parameters really help you there. But I think that's when going into the dose duration approach can be particularly helpful. The only other comments I might add were that when we talked about the solubility modifiers, we talked about both using OLETH and using BSA. But some of the kind of competing considerations were the analytical method. Well, even practically speaking, while Bernau and Stewart did demonstrate that up to 6% BSA did not affect the water permeability constant, and they also looked at cortisone and urea and other things, I do not recommend using 6%. You don't want to use 6% in most situations. There usually isn't a dramatic advantage in solubility enhancement beyond about 0.5%. So you typically start with 0.1, you can go to 0.2. With some very hydrophobic drugs, you can try to go at 0.5. I've never gone beyond, much beyond that unless I was really trying things out, and then it didn't work, to be honest. And part of the problem is that your peak shape will really start to lose form if you have higher amounts of the OLETH in there. You can also start to have much more foaming and bubbles and difficulty, and especially with mass spec methods, even if you have protein in there, like if you have BSA in there, you need to make sure that you're not gaining advantage in solubility and then losing advantage in sensitivity because your analytical method now has become more complicated. Also, you will likely need to do some type of sample cleanup, probably some sample extraction, because if you try to just shoot this on a mass spec, you're going to end up junking up your source and causing some other problems as well. So those are some of the considerations that sometimes also that you have to balance as you're finding an ideal. The last thing that I would maybe add to the very nice synopsis that Hiren gave was relating to the skin. Different suppliers of skin have different types of protocols. So there are those that they harvest the skin from organ donors at the same time as eyes or a heart or a kidney would be harvested. The skin is harvested often for transplantation onto burn victims. These are usually done in GTP facilities, good tissue practices. They have trained personnel, they have protocols, they have very consistent methods for dermatoming. It's nice because you get these large sheets of skin, which sometimes from a surgical kind of breast reduction or abdominoplasty, you don't get very uniform or large sheets of skin. And often those are even cryopreserved. But other sources of skin you might get from a whole body donation facility here, unlike the organ donors who really can be in a range in age from their 20s to their 70s or 80s. Sometimes they're gunshot wound victims or motorcycle accident. The otherwise healthy skin. The whole body donation facilities can sometimes be very elderly people. The state of their skin can be a little bit different. Sometimes you have very little understanding of how much time there was between the time the person passed and the time that the skin was harvested, and what the body, what conditions the body was stored under during that time, or even handling considerations, or even experience of the person harvesting the skin, because the whole body donation facilities, they don't have expertise necessarily in dermatoming, for example. So these can be some of the considerations. And one of the questions was, well, let's see that it is most reliable to be able to get skin from the latter source where the majority of your population is, let's say, 80-year-olds. Is that a problem? And I think the real answer to that will come from being able to use the barrier integrity test to tell you, do we have a competent barrier on these donors? And two, are you able to get a reproducible, precise, discriminating IVPT method? If you are, then that tells you your skin is good. And in fact, the barrier properties of the skin are so critical that we typically need to have them in order to keep living. And so it's not necessarily a problem to be able to have skin from older individuals, but the procedures that you go through in the validation of the IVPT will in fact validate whether or not there were any issues there. So I think that concludes then both our synopsis, and a big thank you to Ying also, who helped to lead the. Ying, did I miss anything?
This definitely shows how the systematic efforts look like in the method development and even validation and subsequent studies. I would like to highly recommend to provide a detailed and well-organized reports in your under submission, because that's what the assessors will be able to see. And then they are hoping they can reconstruct what you have done, what have you been thinking during your development and subsequent studies. Thank you.
Yeah, I think that's a very good point. We understand, we know that it's an exploratory process. You don't always know where you're going to go next, but you should know what you've just done. You should be able to have a record of that, and you should be able to reconstruct how you moved through the process and how you got from one place to the next. And a lot of that information really be really highly to your advantage to be able to record and report on that process. So with that, I'll say thank you to everyone. We'll close this aspect, and we're going to move into our encore session.